The relentless progression of human aging is inextricably linked to a gradual, often imperceptible decline in skeletal muscle mass, strength, and functional capacity, a complex physiological phenomenon universally recognized within the medical and scientific communities as sarcopenia. For decades, the creeping fragility associated with growing older was accepted merely as an inevitable consequence of the passage of time, a biological destiny that awaited everyone who lived long enough to experience it. However, modern scientific inquiry into gerontology, metabolic endocrinology, and exercise physiology has fundamentally dismantled this defeatist paradigm, revealing instead that age-related muscle wasting is not a strictly immutable reality, but rather a highly modifiable condition driven largely by an insidious physiological state known as anabolic resistance. Anabolic resistance characterizes a blunted, inadequate response of aged skeletal muscle to the primary stimuli of muscle protein synthesis, which are dietary protein ingestion and mechanical loading through physical activity. In a youthful physiology, a nominal dose of dietary amino acids, coupled with moderate physical exertion, is sufficient to trigger a robust anabolic cascade, seamlessly repairing and expanding the myofibrillar network. Conversely, in the aging body, the cellular machinery responsible for this synthetic process becomes sluggish, deafened to the biochemical signals that once initiated rapid growth and repair, requiring significantly higher thresholds of both mechanical tension and nutritional intervention to ignite the exact same molecular pathways. To conquer this formidable barrier and reclaim physical vitality, we must engineer a profound paradigm shift in our approach to geriatric nutrition and exercise, moving away from generalized advice and toward hyper-targeted, chronobiological interventions. This brings us to the core thesis of our strategy: the absolute necessity of bridging the anabolic resistance gap by synchronizing the ingestion of leucine-rich protein within a precise, non-negotiable 15-minute window immediately following physical exertion. This specific temporal intervention acts as a potent metabolic lever, an acute biophysical hack designed to exploit a fleeting physiological state where the aging muscle is transiently sensitized to exogenous amino acids, thereby maximizing myofibrillar protein synthesis and effectively halting the progression of age-related muscle atrophy. To truly comprehend the profound impact of this 15-minute protocol, we must embark on a detailed, microscopic journey, visualizing these invisible processes through the lens of a highly detailed three-dimensional cellular animation, a conceptual tool that will vividly illustrate exactly how this precise timing transforms ordinary, routine physical activities into incredibly powerful catalysts for long-term physical independence. Imagine a cinematic, three-dimensional cellular animation beginning at the macroscopic level of an aging human body engaging in a routine physical activity, such as standing up from a chair repeatedly or carrying moderately heavy groceries up a flight of stairs. While these activities may seem mundane, they represent a critical mechanical stressor, a form of resistance training that generates tension across the skeletal muscle fibers, particularly the fast-twitch type 2 fibers that are disproportionately targeted and decimated by sarcopenia. As the animation zooms in, transcending the skin and connective tissue to reveal the architecture of the muscle belly, we observe the initial critical phase of mechanotransduction, the biological process by which mechanical forces are converted into biochemical signals. The physical stretching and contracting of the muscle fibers physically deform the sarcolemma, the cell membrane of the muscle fiber, stretching specialized mechanosensory proteins known as integrins. These integrins, acting as biochemical antennas, immediately transmit this physical tension inward, activating focal adhesion kinase and subsequently stimulating the release of intracellular calcium. Simultaneously, the exertion triggers a localized vascular response. The microscopic capillary beds surrounding the working muscle fibers dilate rapidly, a phenomenon known as exercise-induced hyperemia, driven by the endothelial release of nitric oxide. Blood rushes into the tissue, swelling the interstitial spaces and creating a localized environment primed for nutrient delivery. However, and this is the critical juncture where anabolic resistance typically thwarts the aging individual, this hyperemic response and the corresponding mechanical sensitization of the muscle cell are incredibly short-lived in the elderly. The aged vascular system, often compromised by arterial stiffness and reduced endothelial function, cannot sustain this augmented blood flow for the prolonged periods seen in youth. The window of opportunity is not the generous 1 or 2 hours often cited in traditional sports nutrition literature. In the context of overcoming elderly anabolic resistance, this window snaps shut with ruthless efficiency. This is exactly why the 15-minute temporal deadline is the absolute linchpin of the entire strategy. If the nutritional payload does not arrive while the cellular antennas are vibrating with mechanical tension, and the capillary beds are engorged with blood, the anabolic potential of the exercise is permanently lost, dissipating into the ether of metabolic inefficiency. To capitalize on this incredibly narrow window, the precise nutritional payload must be introduced almost concurrently with the cessation of exercise. The animation shifts to the digestive tract, visualizing the ingestion of a rapidly absorbing, high-quality protein source, specifically one heavily fortified with the essential amino acid leucine, such as a high-grade whey protein isolate. The choice of a liquid isolate over whole food protein sources like meat or eggs is not a matter of mere convenience, but a strict biochemical necessity dictated by the 15-minute time frame. Whole foods require extensive mastication, gastric churning, and prolonged enzymatic breakdown, a process that takes hours, rendering them entirely useless for the immediate post-workout rescue mission required by the aged, anabolically resistant muscle. Furthermore, aging is frequently accompanied by a decrease in the production of stomach acid, a condition known as hypochlorhydria, which further decelerates the digestive process. A liquid whey isolate, however, bypasses these gerontological digestive hurdles. As the animation tracks the fluid entering the stomach, we see it rapidly empty into the duodenum, where pancreatic proteases swiftly cleave the predigested peptide bonds. Within mere minutes, a torrential flood of individual amino acids, championed by a massive surge of leucine, is transported across the intestinal epithelium via specialized peptide-1 transporters, plunging directly into the portal vein and entering systemic circulation. Because the protein was consumed immediately within that 15-minute window, this massive amino acid spike coincides perfectly with the lingering exercise-induced hyperemia in the peripheral musculature. The animation follows the leucine molecules as they are pumped through the arterial system, arriving at the exact capillary beds that were dilated by the physical exertion just moments prior. The timing is a perfectly synchronized physiological ballet. The doors of the muscle cell are wide open, and the nutritional building blocks have arrived exactly on time. As the leucine molecules exit the capillaries and enter the interstitial fluid bathing the muscle fibers, the true molecular magic of this intervention begins to unfold on our three-dimensional stage. To cross the sarcolemma and enter the intracellular matrix, leucine utilizes a specific bidirectional transporter known as the L-type amino acid transporter 1, or LAT1. Interestingly, the mechanical tension generated by the exercise just minutes before has actively upregulated the density and efficiency of these LAT1 transporters on the cell surface, pulling the leucine into the cell with an aggressive affinity that would not exist in a sedentary state. Once inside the sarcoplasm, leucine ceases to be merely a structural building block for tissue. It fundamentally transforms into a highly potent signaling molecule, a biological key designed to unlock the master regulator of muscle protein synthesis, the mechanistic target of rapamycin complex 1, universally known as mTORC1. In the context of anabolic resistance, the aged mTORC1 pathway is notoriously stubborn, heavily suppressed by age-related chronic low-grade systemic inflammation, often referred to as inflammaging, as well as an accumulation of reactive oxygen species resulting from mitochondrial dysfunction. To overcome this suppression, a gentle nudge is insufficient. A sledgehammer is required. This sledgehammer is the acute, massive intracellular spike in leucine concentration. The animation zooms down to the nanometer scale, revealing the complex protein machinery within the sarcoplasm. We witness leucine binding to a specific intracellular sensor protein called cestrin-2. In a fasted or low-protein state, cestrin-2 acts as an inhibitor, binding to and silencing a complex known as gator-2, which indirectly keeps mTORC1 in a dormant, inactive state. However, when the massive influx of leucine binds to sestrin-2, it induces a dramatic conformational change, forcing sestrin-2 to release GATOR-2. This single elegant molecular uncoupling sets off a cascading domino effect. The liberated GATOR-2 inhibits GATOR-1, which in turn allows a family of proteins called the RAG-GT-PASEs to become active. These active RAG proteins physically grab the dormant mTORC1 molecule and forcefully translocate it to the surface of the lysosome, a cellular organelle. Waiting at the lysosomal surface is another protein called Reb, which, crucially, has been simultaneously activated by the mechanical tension of the prior exercise via the phosphatidylinositol 3 kinase, PI3K, and protein kinase B ACT signaling pathway. It is here, at the lysosomal membrane within the strict 15-minute window, that the ultimate convergence occurs. The nutritional signal of leucine and the mechanical signal of exercise meet, allowing Reb to bind to and fully, explosively activate mTORC1. With mTORC1 fully activated, the resistance of the aged cell is officially broken, and the process of myofibrillar protein synthesis is violently initiated. The animation portrays this activation as a brilliant flash of bioluminescent light spreading through the muscle fiber, a visual metaphor for the sudden awakening of the cell's synthetic machinery. Activated mTORC1 acts as an elite molecular architect, immediately dispatching chemical messengers to the ribosomes, the microscopic protein factories of the cell. It phosphorylates and thereby activates two critical downstream targets: ribosomal protein S6 kinase beta, 1p70S6K, and eukaryotic translation initiation factor 4E binding protein 1, 4EBP1. The phosphorylation of 4EBP1 forces it to release eukaryotic translation initiation factor 4IF4E, which is then free to bind to the messenger RNA mRNA that carries the genetic blueprints for new muscle tissue. Simultaneously, the activated P70S6K upregulates ribosomal biogenesis, essentially building more factories to handle the incoming workload. We witness the ribosomes docking onto the mRNA strands, reading the genetic code, and systematically pulling in the other essential amino acids that arrive with the leucine. With mesmerizing speed and precision, the ribosomes link these amino acids together, forming long, complex polypeptide chains. These newly synthesized chains undergo intricate folding processes, supervised by chaperone proteins, ultimately maturing into functional actin and myosin filaments, the fundamental contractile proteins of skeletal muscle. The animation shows these new filaments being seamlessly integrated into the existing sarcomeres, the microscopic contractile units of the muscle fiber. Layer by layer, protein by protein, the myofibrils become thicker, denser, and stronger. This is the precise cellular mechanism of muscle hypertrophy and repair. By forcefully driving this process through the synchronized 15-minute leucine and exercise protocol, we are not merely stopping the progression of sarcopenia, we are actively reversing it, rebuilding the structural integrity of the aging muscle fiber from the inside out. Furthermore, this intense anabolic signaling also stimulates muscle satellite cells, the dormant stem cells residing on the periphery of the muscle fibers. The activation of mTORC1 prompts these satellite cells to proliferate and donate their nuclei to the existing muscle fibers, increasing the cell's overall myonuclear domain and permanently enhancing its capacity for future protein synthesis. This effectively means that the 15-minute protocol not only builds muscle today, but physically upgrades the cellular machinery to make building muscle easier tomorrow, systematically eroding the foundation of anabolic resistance. The profound physiological transformations occurring at the microscopic level cascade upward, generating a massive paradigm shift in the macroscopic reality of the aging individual, fundamentally altering their psychological landscape and their behavioral relationship with their own body. To understand the gravity of this lifestyle integration, one must first deeply analyze the profound psychological toll exacted by unchecked sarcopenia. As muscle mass silently wastes away, so too does physical self-efficacy, a psychological concept defined as an individual's belief in their innate ability to execute behaviors necessary to produce specific performance attainments. When an elderly individual struggles to rise from a low chair or feels unsteady navigating a curb, a subtle yet devastating psychological trauma occurs. A pervasive, insidious fear of falling takes root, leading to a phenomenon known as fear avoidance behavior. The individual subconsciously begins to restrict their physical activity, avoiding stairs, declining social invitations that require walking, and generally shrinking the physical perimeter of their life. This reduction in activity accelerates the loss of muscle mass, which in turn breeds more frailty and more fear, creating a vicious, downward psychosomatic spiral that ultimately culminates in a complete loss of physical independence and reliance on external care. The 15-minute protein synchronization strategy represents a radical psychological intervention designed to shatter this learned helplessness. By establishing a strict, non-negotiable protocol, the immediate consumption of the leucine-rich beverage following any strenuous routine activity, the elderly individual is granted a highly specific, actionable locus of control over their physical destiny. It transforms the nebulous, overwhelming goal of getting healthier into a concrete, daily ritual. When the individual finishes carrying the laundry up the stairs or completes a session of chair squats, they are no longer merely tired. They are acutely aware that they have primed a biological pump, and they have exactly 15 minutes to pull the lever. This creates a powerful sense of agency and purpose. The protein shake is no longer just a dietary supplement. It is conceptualized as an immediate, life-saving medicine, a tangible reward for their mechanical exertion. This ritualistic coupling of effort and immediate nutritional rescue rewires the cognitive behavioral pathways. The individual begins to view their daily physical struggles not as reminders of their decaying physical state, but as deliberate opportunities to stimulate mechanotransduction and activate mTORC1. The heavy groceries become dumbbells, the staircase becomes a leg press. Because the biological response is so intensely amplified by the precise timing, the physical results manifest much more rapidly than with disorganized, generalized exercise and diet programs. Within a matter of weeks, the individual notices a tangible increase in their kinetic chain stability. The chair that was once a trap becomes easy to rise from. The balance that was once precarious becomes solid and reliable. As these physical capabilities return, the fear avoidance behaviors dissolve. The psychological perimeter of their life expands once again. They begin to actively seek out physical challenges, confident in their newly acquired knowledge that as long as they respect the 15-minute post-exertion window, their body will respond not with injury and fatigue, but with regeneration and strength. Beyond the immediate mechanics of myofibrillar protein synthesis and the profound psychological restructuring, the strict adherence to this 15-minute leucine synchronization protocol initiates a broader, cascading symphony of systemic metabolic improvements that drastically enhance overall longevity and disease resistance in the elderly. Skeletal muscle is far more than mere mechanical rigging for the skeleton. It is recognized by modern endocrinology as the largest endocrine organ in the human body. When skeletal muscle contracts during the mechanical loading phase of our protocol, it secretes a vast array of signaling proteins known as myokines, most notably interleukin-6 (IL-6), which exert powerful systemic anti-inflammatory effects, actively combating the destructive systemic inflammaging that accelerates all major age-related diseases, including neurodegeneration and cardiovascular disease. By repeatedly preserving and expanding the muscle mass through our targeted post-workout nutritional intervention, we are effectively increasing the size and output of this crucial endocrine gland, bathing the brain, heart, and internal organs in protective, anti-inflammatory myokines. Furthermore, skeletal muscle serves as the primary reservoir for glucose disposal in the body. Age-related sarcopenia is inextricably linked to the development of age-related insulin resistance and type 2 diabetes, simply because the body loses the physical tissue required to absorb and store glucose from the bloodstream. By reversing muscle atrophy through the precise activation of mTORC1, we significantly increase the expression of GLUT4 transporters on the expanded muscle cell membranes. When the individual consumes carbohydrates, the glucose is now rapidly shuttled out of the bloodstream and into the newly constructed muscle tissue, stabilizing blood sugar levels, reducing the pancreatic burden of insulin production and drastically lowering the risk of metabolic syndrome. Furthermore, the biomechanical tension generated by the routine activities used to trigger the 15-minute window exerts a profoundly beneficial effect on bone mineral density. The physical pulling of the enlarged, strengthened tendons on the skeletal structure creates microstrains within the bone matrix, stimulating osteoblast activity and actively combating osteopenia and osteoporosis, two conditions that run aggressively parallel to sarcopenia. The structural fortification of the bone, combined with the dramatically improved balance, proprioception, and reactive force generation provided by the newly synthesized type 2 muscle fibers, practically eliminates the risk of catastrophic falls, which are frequently the precipitating event for terminal decline in the elderly population. The implementation of this strategy requires a meticulous understanding of nutritional formulation, specifically tailored to the unique physiological limitations of the aging digestive and metabolic systems. It is not sufficient to simply consume any protein source within the 15-minute window. The biochemical profile of the intervention must be mathematically precise. Leucine is the trigger, the absolute necessity for overcoming the anabolic resistance threshold, and research indicates that the aged muscle requires approximately 3 to 4 grams of pure leucine per serving to achieve maximal mTORC1 activation, a threshold nearly double that required by a younger individual. To obtain this much leucine from a whole food source like chicken or beef would require consuming an uncomfortably large portion, which is highly impractical for an elderly individual with a diminished appetite, a common gerontological condition known as the anorexia of aging. Moreover, as previously established, the delayed gastric emptying of solid food would completely miss the critical 15-minute window of heightened microvascular perfusion. Therefore, a highly refined whey protein isolate, specifically one artificially enriched with supplementary free-form L-leucine, becomes the scientifically mandated delivery vehicle. The isolate processing removes the fats and lactose, ensuring that the liquid passes through the gastric sphincter almost instantaneously. However, triggering mTORC1 with leucine is merely the initiation of the construction project. It is the hiring of the contractor. To actually build the myofibrillar tissue, the ribosomes require all 9 essential amino acids to be present in the sarcoplasm in adequate quantities. If leucine triggers the synthesis, but the body lacks sufficient valine, isoleucine, lysine, or methionine, the synthesis process will prematurely terminate, a phenomenon known as the muscle full effect, severely limiting the hypertrophic response. Therefore, the post-workout beverage must be a complete spectrum of essential amino acids, heavily biased toward leucine, dissolved in a fluid that ensures rapid hydration and electrolyte replenishment, as cellular hydration status directly influences the volume of the muscle cell and independently stimulates protein synthesis. This highly engineered liquid payload must be prepared in advance, waiting on the kitchen counter or in the refrigerator, completely ready for immediate consumption the exact second the physical exertion concludes. The individual cannot waste the critical 15 minutes preparing the drink, searching for ingredients, or waiting for a meal to cook. The biological timer starts ticking the moment the mechanical tension ceases, and the shrinking capillary beds are the physical embodiment of the closing window of opportunity. When we integrate the complex molecular biology of the mTORC1 pathway, the biophysics of mechanotransduction, the chronobiology of post-exercise microvascular perfusion, the precise nutritional kinetics of leucine-enriched whey isolate, and the profound psychological implications of reclaiming physical agency, we construct a comprehensive, unbreakable, and scientifically rigorous bulwark against the ravages of aging. We are not merely trying to slow down an inevitable decline. We are aggressively intervening at the cellular level to force the biological machinery into an anabolic state that defies its chronological age. We have envisioned, through the lens of our three-dimensional cellular animation, the exact mechanism by which a mundane physical effort, lifting a grandchild, working in the garden, or rising from a deep armchair, can be instantaneously converted into a massive biological signal for growth, provided that effort is immediately married to the precise influx of amino acids. This 15-minute strategy elevates the concept of aging gracefully from a passive, defensive posture into an active, offensive campaign for bodily autonomy. It demands a level of discipline and chronobiological awareness that transcends casual health advice, establishing a profound, daily ritual wherein the elderly individual consciously overrides their innate anabolic resistance through the sheer force of applied biochemical timing. By viewing their daily routine not as a series of tiring obligations, but as a deliberate sequence of mechanical triggers designed to prime the muscle cell, and by treating the subsequent 15-minute window as a sacred, non-negotiable period of nutritional rescue, the aging individual systematically reconstructs their physical foundation. The muscle fibers thicken, the intracellular factories multiply, the bone density stabilizes, the metabolic glucose sinks expand, and the psychological shadows of fear and frailty are permanently banished by the undeniable, physical proof of their own enduring strength. This intricate, highly specific, and scientifically absolute synchronization of effort and nutrition is the ultimate key to unlocking a vibrant, functional, and fiercely independent longevity, fundamentally proving that while we cannot stop the clock, we absolutely possess the power to dictate exactly how our bodies respond to the passage of every single second.